I'm actually subbing in for Greg Jeffries. Can I get a show of hands who's seen Greg talk before? Right, good. So most of you don't know what you're losing out on. Okay, good. So this is the overview. As a disclaimer, while I was listening to the talks this morning, which were all great, I ended up sort of swapping things out and then just sort of tuning to the audience. So let's see how this goes. But broadly speaking, 3 parts. The first one is just sort of a bit of advertisement for the newest addition to the set of FlyWire, Fly Connectomes. That's a project that was a collaboration between labs in Princeton and us in Cambridge and a whole range of other people. And I think I'm the only one, no, not the only one, but certainly the first one to talk about it. So I'm going to fly the flag a little and advertise. Then the second one is the, sort of the meat, lessons learned from comparing multiple connectomes, 2 connectomes. And then I'm going to briefly dip into cell typing. Okay, so the previous talks have already done a lot of heavy lifting in terms of introducing FlyBrains and EM data and connectomes, but I'll sort of try to fill in bits and pieces here and there. So for those of you who've never seen a FlyBrain, that's what this looks like. So you can, you can imagine it sitting in the head capsule between the eyes. So that's the actual central brain. The structure and outline in yellow is the central brain, and then to the left and right you've got the optic lobes, and the eyes then sort of sit on top of this. And then below that you can imagine there being sort of a thick nerve that connects the brain to the nerve cord, which is the spinal cord equivalent. And the whole thing is about 1 millimeter across, not quite as tall and rather thin, so about the size of a poppy seed. And for those of you who've never worked with insect neurons, or fly neurons specifically, but with, say, vertebrate neurons, that's a 2-scale example comparison of a neuron from the cortical microns dataset from the Allen, i.e. a mouse neuron in teal, and an actual fly neuron from a connectome, the hemibrain in this case, in purple. And so this is just to sort of give you an idea of the scale, right? Like very small brain, the neurons aren't actually that much smaller, but there they are. Okay, so now why do we care about FlyBrains in the first place? I think we've already heard good reasons in the talks this morning, but just to sort of throw a couple more interesting systems or behaviors at you. So the FlyBrain, despite its small size, we're looking at about 140,000 neurons in this tiny thing, is capable of very complex behavior, and interesting behavior at that. So, we've already heard about navigation from Vivek this morning. It is capable, the fly is capable of associative learning memory. People study olfactory processing, innate behavior, as sort of, in contrast to the learned behavior. Visual processing, I think Srini might talk about this. Are you on tomorrow?
Wednesday.
Wednesday, all right, so something to look forward to. And by the way, Sang-Soo
He's coming here later tomorrow.
Okay, so Sang-Soo might also talk about visual processing. But we've also got motor control, motor sequences, decision-making, sensory integration, sexual-morphic behavior, and all these, lots of other interesting things. And you can see that actually quite a few people, those that are underlined, who work on these things are in the audience and give talks. So, keep an eye out. Okay, so we've got a small brain. It's, tractable. We've got nice behavior. Interesting circuitry. The other sort of, I guess you could call killer app is the fact that it's genetically tractable. I guess this is actually where Drosophila comes from, right? Originally, it was primarily a genetic model organism. And this is just an example of that. This is sort of the tip of the iceberg, if you will. It's a publication from just a couple of weeks ago from the lab of Jerry Rubin at Janelia, where they published this resource of over 3,000 genetic driver lines. And you can see in these images, they look pretty much like what Vivek showed earlier. It essentially gives you genetic access to very specific populations of neurons down to the single cell type. So that lets you then, in this case, they express GFP, right? So you can image those neurons, but you can imagine that you can use this to optogenetically activate or inactivate those neurons, or to use calcium imaging to track the activity. So basically, you can drill down really well. Okay, and then in the context of this conference, the other thing that's obviously relevant is that it's small enough that you can do EM connectomics on it. I think, pretty obvious. I thought I'd still say it. So this is 2-scale, the FlyBrain, and an artistic rendering of the cortical microns data set. So this is, as far as I know, still the biggest EM data set that we have. So it's, a millimeter cubed. Sorry, yeah, a millimeter cubed. You can fit 50 FlyBrains in that volume. Okay, we already had a connectome, or we already have a connectome from around 2020, the Geneva hemibrain connectome. So you can see it covers about half a brain, the bottom part, and most of the optic lobe is cut off. But we already had half a brain, give or take. 22,000 neurons, about 5,000 cell types. Okay. Now, obviously, I want to talk about comparative connectomics, so we need a second one, and this is where the FlyWire connectome comes in, and I'm just going to give you like a brief history because I think those tidbits are interesting, and maybe make you remember this in the future. So what you see here is the FAFB image volume. That's an EM image volume of a full adult FlyBrain. It was actually imaged in 2015 already. And what we're seeing here is sort of like all zoomed out, and we're going anterior to posterior slice-wise. The resolution is high. 4 by 4 by 4 nanometers, so we could zoom in and look at individual synapses. Now, so as I said, this was imaged by Zhihao, who was a postdoc with Davi Bock at the time at Geneva between 2015 and 2016. And then for a long time, researchers from all over the place, including our group, were manually reconstructing in there. So you can imagine sort of, this web-based software called CatMate, and we sort of traced neurons, annotated synapses, and everyone was doing their sort of small circuit of interest. Took forever. In hindsight, horrendously slow. Can't recommend it. And I should say that, this was like, 4.5 years. At the end of it, based on cable, we reconstructed about 10% of the brain, and that's probably that's being generous. Right, so that's as far as we got. Now, in comes Sebastian. So the reason why this volume was never segmented is because it's a serial section EM volume, right? So essentially you slice the brain, put it on wafers, on grids, and then you image them, and you have to reassemble it in silico. Now that introduces alignment issues, which at the time people thought, you can't segment this, right? Like you can't bridge these gaps and these sort of shifts. In comes Sebastian and some people in his lab. They used a neural network to essentially fix those alignments and then segment the brain. So essentially they, turned this manual reconstruction into something automatic that needs proofreading. And this is where, I should say, I remember we talked about this earlier, I think. There was the Berlin Connectomics conference in 2019 when Sebastian, sort of fresh off the print, showed us the first results of these neurons, right? So basically showed us volume, clicked on a couple of neurons, and boom, they showed up. Very exciting. The problem then is that you still need to fix up these volumes, right? I think we've briefly heard about proofreading before, right? Essentially the computer makes mistakes and you need a human proofreader to fix up those neurons. So this is a huge undertaking. And Sebastian's lab had experience. Have you heard of IWire before? It's a sort of gamified reconstruction in the visual cortex of, is it retina? Retina in a mouse. Anyway, so they set up a similar system in collaboration with the lab of Mala Murthy and spearheaded by Sven Dorkenwald, where they invited researchers, but also citizen scientists, and there were a bunch of paid tracers too, to proofread in this data set. And so over the course of the next 4 years, that was done. I'll show a timescale of this in a second. And then so we released the first version of this at the middle of 2023 that covered the central brain and a good chunk of the optic lobes. And as of, 3 weeks ago, I think, we've released the second version of this, which now includes everything. So that exists now. Just briefly, what you see on the right is an animation of the edits that were made. In total, over 3.1 million edits. You can see sort of over time how the brain pops up. the number of proof-read neurons on the right. The important thing is that if you tally this up, Sven did that, you end up with 30 person years to proofread this data set. What do you get for it? You get this. That's the FlyWire data set. So essentially, the way insect brains work is you've got all the cell bodies on the outside in the cortex, and then the neurites are on the inside. So essentially what we're seeing is sort of this layer, this cell body rind, basically. All right, I already said 3.1 million edits, 30 person years, about 1/50th of a cubic millimeter, which is 1/25,000th of a mouse brain, right? This is how far we're still away from a mouse connectome. 139,000 neurons, about 150 meters of cable packed into this poppy seed-sized brain, and 130 million synapses. On top of that, we also spent about 3 person years annotating the data set. So essentially, right, if you don't label these things, then all you give people is sort of, ID X, ID neuron 1 connects to neuron 2 with 3 synapses, which is fine if you know the data set, then you can sort of keep track of things. But if you want the wider public, the community to use it, you have to make it parsable. And so we did that.
What's an example of one of the proofreading corrections?
Okay, so proofreading corrections is basically either a split or a merge, right? So you've got a neuron, and there's a bit that shouldn't belong to that neuron, you cut it off. Or the segmentation missed a branch, and you have to attach it. Those two, basically, this is the two processes, 2 kinds of edits you make.
The annotation is similar to hemibrain or different?
Sort of. You can see that we actually have quite a few more, which is because we kind of had this hierarchy, started with this hierarchy of annotations, because in a hemibrain, you had mostly cell types, and not all people actually care about the cell types, so we have some higher, sort of coarser granularity with that. It's just, hopefully making it more easy. And this is actually, incidentally, this is illustrating. This is the coarsest granularity. So here we're rendering superclasses. The first one is the central superclass. Now we're looking at all the descending neurons, right? So these are neurons that take information in from the brain and bring it to the nerve cord, so the spinal cord equivalent. We've got motor neurons. There aren't that many in the brain. Most of them sit in the spinal cord equivalent. Sensory neurons, right? So whatever, visual system, olfaction, gustation, all that kind of stuff. What's next? Endocrine neurons. Those are neurons that receive input in the brain and release hormones into the hemolymph. Ascending neurons, so basically the opposite of descending neurons. So these communicate between the nerve cord and the brain. And then we have the visual system. That's the visual system, and then you've got neurons that connect the optic lobes with the central brain, i.e. the visual centrifugal neurons, so they go from the brain to the optic lobes, and last but not least, might as well wait for the last one, visual projection neurons. Yes?
And this is conserved across animals?
Yes, I mean, this coarse structure?
Yes, at
yes.
this level of resolution.
Yeah,
Yes.
I'm like that. That level of resolution is pretty straightforward. Basically, once you go into the terminal cell type, this is where it gets a bit more tricky, but I'll cover that too, I hope. We get to that. All right, so for you, where can you get the data? Your first port of entry should probably be codex.flywire.ai. That's a platform developed by Ari from, I think he's technically in Mala's lab, but might be Sebastian too. So it's essentially kind of like Newprint, but for FlyWire, if you've heard of that before. The important thing is you can use it you need a Google account to sign up, no problem there. And then you can explore interactively, just sort of search for neurons, look up connectivity, but you can also, importantly, you can just download the data, right? So if you just go there and you want the connectivity, the edge list basically, and the annotation separately, it's all there.
So this connectivity is like equivalent to like a matrix?
No, that would be, basically it's sparse, so you get an edge list. Alternatively, we've also developed R and Python packages that just happen to be both called FAFBSeg, that you can use to programmatically interact with the data sets. Okay, so now that we have this second connect.
Super cool. Is there also neurotransmitter annotation?
Yes.
What? Okay.
Actually, the neurotransmitter predictions were originally done on FAFB, which is the same image volume as used for FlyWire, so that's the original prediction data. Right, so what can you do with this? now we're getting into the biology. One of the things that we're interested in, and I guess the talk title gave it away, is how stereotyped are brains. And sort of implicit in that is this sort of notion, this sort of question that the field of connectomics has to contend with occasionally. It's kind of like asking, so you've got an N of 1, or maybe 1.5 now. How representative is this one brain for all the other brains of the species, right? Like, or put it in a different way, have we collected a snowflake, right? Like, it's beautiful, but it's so unique, it doesn't tell you much about the next snowflake. So that's the kind of question we would like to address. And in order to do that, we need a mapping. So we need to basically be able to say which neuron in the hemibrain corresponds to which neuron on the left-hand side of FlyWire, say, right? And pairwise then. Now, there's many ways of doing this. For us in practice, what it boiled down to is using morphology. I think, for which of the two of you mentioned this? I think Ashok may have mentioned that fly neurons are super stereotyped, right? Like basically you can find the same neuron over and over again in pretty much every brain. So morphology is actually a very strong metric, at least, well, depending on the metric, but it's a good start. And then obviously you can use connectivity, which in our case usually ends up being we use morphology for the coarse matching, and then if we need to refine, we use connectivity. Now in practice, the pipeline looks something like this, and I'm going to go into a bit of detail because I think that might be interesting. But basically, we take all the hemibrain neurons, we transform them using a non-rigid transform into FlyWire space, run an NBLAST. I'll explain what that is later. But effectively, this gives us a similarity matrix, and then we can figure out which neurons match. Right. So this illustrates the spatial transform. Right. So on the left-hand side, you see the hemibrain volume. I hope you can see that. It's a bit faint. And on the right is the FlyWire brain, and it's tilted. That's because X and Z-axis Y and Z-axis are swapped between the two data sets. And you can see that this is an absolute space. And what we're doing here now, once I press play, we're going to transform the red neuron that's in the hemibrain space into FlyWire. And at the same time, look at the axis. We're also moving the camera slightly. Right? So essentially, that's the spatial transform. And I hope you can appreciate that we're able to put the neuron in the right place in the other data set. And we just do this with all the hemibrain neurons. And then we run NBLAST. Just briefly, NBLAST essentially treats neurons like point clouds. And each point has a tangent vector that indicates the directionality of the neuron. And then what you do is, if you've got a query in a target pair, right, so it's a pairwise query, you essentially run a nearest neighbor lookup. And then there's a scoring function that takes both the distance between the point pairs and the dot product of the vector into account. And then the total score is effectively the sum over all the individual scores. And usually that's normalized to a self-hit. So 1 means self-hit, and then it goes all the way to negative, right? So you can see that the scoring function actually goes negative. Okay, I'm just going to briefly mention this because I think it's an important point, but I don't think I can make that point without giving you a bit of the data. Okay, so what you see here is for every single hemibrain neuron, the top NBLAST score in the FlyWire data set. So the question here is like, which of these are actually plausible hits? Empirically, we know that everything above 0.3 is probably decent, and everything above 0.5 is safe bet. So to sort of figure this out a bit more quantitatively, we essentially sampled these scores in the different bins and then scored yay or nay. And what this comes down to is if you take our sort of manual scoring of the sample and effectively multiply this with the distribution of scores, 99% of neurons in the hemibrain have a good match in FlyWire, which is exactly the point that Ashok was making earlier, right? Like for almost all neurons, there's something that looks like it. Now, for our mapping, ideally you get something like this, right? So you've got a FlyWire neuron, you've got a hemibrain neuron, and they're each other's best match. The reality is that it's more often than not looks like this, right? So you're kind of like, well, it could be this, could be that, or even worse, right? Like could be this, could be that, but then it might also be this. So you get these sort of more connected components, so almost, yeah?
You were asked earlier what kind of mistakes did people correct when they did it, looking by hand. Would the lack of matches in the previous slide guide you to where to look for those mistakes?
That's a good...
Go back to the cases that didn't match and look more carefully there?
Yes, that's exactly what happened. So effectively, this kind of analysis, this kind of matching, we did pretty early on in the proofreading process. And then you're right, if you don't find a match, then the first thing you want to do is figure out whether there's something wrong with the neuron and not with the data. It's in parts complicated by the fact that the hemibrain is truncated, right? So it's just half a brain, and neurons dip in and out, and sometimes, well, a lot of the neurons actually only partially contained in that volume. But yeah.
So, you could just give us some intuition. I mean, you said that 0.3 is good, 0.5 is wonderful. I mean, but what is the intuition for these numbers? I mean, if there weren't exactly matching neurons between the two flies, what would we get? I mean,
Good. I mean, I don't have a slide with an example, unfortunately. It's a bit of a spectrum, right? So we definitely found like a handful, less than 10 neurons, where we can clearly say these neurons exist on the left side of FlyWire and on the right. So we actually have them on both sides of the same brain, but they definitely don't exist in the hemibrain. And then there's sort of variations on this, right? Where like, in FlyWire, these neurons make a turn here, they don't do this in hemibrain, and the question is like, is this still the same cell type? You can look at the connectivity, maybe it doesn't make it, like the morphology doesn't affect the connectivity, some compensation going on. So again, it's a spectrum. We've been very conservative with this stuff. So essentially, if it's even slightly off, it would have been among the 1%. So if you give it a bit more wiggle room, we probably could have pushed us even higher and said, 99.9% have a match.
I think I'm asking a much more naive question than the one that you're trying to answer. When should we declare victory with your measure from NBLAST or whatever it's called? Is 0.5 the magical number because you can overlay the dendritic tree to the level and say, look, these are two identical brothers, and we will never tell them apart. I mean, I'm not arguing about, just trying to get a sense of when are you happy?
Yeah, that's a good call. So I should say that this, and also we'll get to the sort of the bigger picture effort, was all basically manually reviewed by a human at some point. So people have to make a judgment call. And sort of the rule of thumb was, if you think that you can tell it apart in light level, right? So you do see small differences. If you look at the same neuron, we know that's the same neuron because it only exists once, it's unique, right? We can say this is the same neuron. But even those neurons that are a safe bet to be the same, there's like small differences, right? Like branch here, branch there. Our rule of thumb was always like, if you think you can tell it apart in light level, then we say it's the same neuron, it's a good match. I'll try over the next couple of days to show you a couple examples. It's minor difference, really. Like, they're really good matches. They're so highly stereotyped. Okay, so we were here, right, where things go crisscross. Now, in this configuration, and let's say we've reviewed those and they're all plausible. I can't tell whether this neuron is this neuron or this neuron is this neuron. Sorry, whether this neuron is this neuron is this neuron, or the other way around. Right? So essentially, you have to treat them as a group. And this is what we say, well, that's the cell type then, right? Because the cell type, yeah?
This degeneracy here, how much of that is because of the hemibrain cropping versus, I know you're now talking about the bigger picture point about cell types. Totally makes sense.
So the truncation really messed with a lot of cell types. You'll see later that we were able to essentially take about 65-67% of the hemibrain cell types and identify them in FlyWire. The remainder, we were just very conservative and said, well, there's something that looks like it, and we could possibly find the same group of neurons, but they're so heavily truncated that we might as well, right? We don't want to ruin our subsequent analyses by including things that we think, might be subpar. So I would say, based on that, just 1/3 maybe, where there was some problem, not necessarily with the truncation. In some cases, it was also just the definition of cell types, but it's a different story. Okay, so cell type, for us at least, is the smallest unit of conservation, both, I guess, in this talk, across brains or hemispheres. And this is an example, right? So this is just an example of one of the hemibrain cell types, or the cell types that were defined originally in the hemibrain. It's called PS008. And we were able to find the same group of neurons on both sides of FlyWire, right? You can see that, for example, here, right, as an example, right? Like, this branch here seems to exist here, but not here. So that's the kind of thing where, we would say that's still plausible. just developmental or biological variability. The important thing is I can tell you that this group is this group is this group. I can't tell you whether this neuron is supposed to be this neuron, this neuron, this neuron, right? That's the whole point I'm trying to make. And now I thought I'd show you this because that's probably for the graph people among you a bit more accessible. So what you see here is each square, green square, is a neuron in FlyWire. Each orange circle is a hemibrain type, essentially just the collapsed neurons representing that type. And the arrows represent the top matches. And the top matches can be just one, but it can also be multiple. And the exercise is essentially trying to figure out what belongs where, right? Or maybe, and that's the complicating factor, maybe the orange dots are wrong, right? Maybe the cell types were wrong. Again, this is sort of a graph representation, and I hope you can appreciate that there's a couple, the ones with asterisks, where they could be matches for both, right? And you have to basically figure out what's what. In some cases, it's obvious, right? N-blast isn't perfect, so sometimes a high N-blast score does not mean that it's the right match. But you know, that's the kind of problem we had when we tried to come up with the mapping between the two of them. We've tried things like bipartite graph matching. We've tried optimal transport. They get you like 80–90% of the way. At the end of the day, you still have to review things if you want to get like high quality matches. If you want to get it as clean as possible. Yeah.
Yeah. So do some of these uncertainties in the mapping reduce when you also put in the additional information that you didn't use so far, which is connections from presynaptic neurons, connections to postsynaptic neurons, maybe based on their match
yes,
with the corresponding partners?
The short answer is yes, and in a lot of cases we actually ended up doing that. You know, you do a morphology match, and then if the morphology match has still some ambiguity, you basically take that subset of neurons and run connectivity only. And often that actually solves it. It's very powerful. The problem with connectivity is you actually need to know some of the labels before you can do that, right? So it's kind of like a circular argument in a way. Anyway, so in total, we were able to match, give or take, 67% of hemibrain cell types. And this is just an example, right? So it's a slideshow, and you see the hemibrain cell type and the FlyWire cell type next to each other in the same brain space. But in total, 3.6 thousand cell types, 15,000 neurons per hemisphere, so left, right, and hemibrain. And the important thing is that they actually cover about 50% of the graph. Okay. What do we get from this? So we've got, with this mapping, we now have 3 graphs: FlyWire right, FlyWire left, and the hemibrain, where each node represents a cell type. So you can see these little stacks, right? So a cell type can be one neuron, but it can be multiple neurons. And the edges are just sort of the summed up edge weights between, the number of synapses between those 2 cell types. Right, and now we can start, I'm going to start simple, right? Like, what if we wanted to know whether this edge exists across all 3 hemispheres, right? So basically, how consistent is that? And if we just ask this very simple question, then we get basically anything between, so somewhere between 50 to 60% of the edges in one brain can be found in another hemisphere. Mind you that obviously, like this is within the same brain, and this is then across. And the numbers are slightly better for within than across. Fine. Yeah?
This tracks with the completion rate of the pre and post, it's the attachment rates of...
Sort of. It's a good, I don't actually, we never try to correlate this specifically. I would have to think about how to do this exactly, but I've got something on that end later. But good point. Okay, so that's binary, right? And essentially, it looks a bit grim, to be honest, right? However, if you then correlate this with the strength of the edge, so the number of synapses, then, somewhat intuitively, right, stronger edges are more likely to be conserved. No surprise there. And just to give you an idea, if you've got an edge of 30 synapses, or made up of 30 synapses, there's a 97% chance that you'll find it in a second brain. Conversely, for one synapse edge, only just under 30%. Also sounds a bit, like slightly pessimistic. But, so first of all, sorry, actually there's something in between I forgot. So we're talking about thresholding earlier. Ashok did. And the consensus seems to be, or sort of people seem to tend to go for 5 synapses. 19,
yeah.
Give or take. Yeah, sometimes 3, right? Like, yeah. But the important thing is that that thresholding is on the level of individual synapses, individual neurons. We're looking at cell types, so thresholds are bound to be slightly higher. So at the level of cell types, we're looking at 11 or more synapses and equal to or more than 0.5% of the target's input. So that's the normalized edge weight. If you use that as a threshold, you've got a 90% chance that that edge can be found in another brain. Again, that's intuitively, that's a high threshold, right? Especially, even if you keep in mind that cell types, it's still a high threshold. However, if you look at the number of, or the fraction of the connectivity, i.e. the number of synapses in the entire graph that is comprised in these above threshold edges, only 16% of edges are above 10 synapses, but they contain about 80% of all synapses. So a lot of the graph actually is contained in the edges that we know are conserved, which I think is encouraging.
I think this is partial absolution for the sin.
Absolutely, yes, yeah. No, totally.
The number.
Ego te absolvo. Okay, so moving on. So far we've looked, it's still a binary question, does it exist or not? What we can also do is ask, okay, so given an edge of 30 synapses in one brain, what's the edge, even if it exists, what's the weight of the edge in the other brain? So that's what we're looking at here. So this is the hemibrain edge weight against the FlyWire edge weight. In this case, on the right-hand hemisphere, looks the same if I use the left-hand hemisphere. The orange line is the average, and then the envelopes are the quantiles. And you can see it's a huge spread, right? Again, to give you an intuition, if you've got an edge of 30 synapses in brain 1, and you look for the same edge across 100 brains, sure, right? Average is 30. But there's a 5% chance that in one of those brains, it'll be 30. It'll be just 3 synapses, which is quite a big contrast. Now that's across brains. We can also do the same thing for within brains, so left versus right, where you would think that some of the issues with the dataset modality, the difference in the dataset, right? Say, the hemibrain, for example, is a FIPSEM volume, presumably less alignment errors. So some of that will go away, and indeed it looks slightly better, but you can see the envelopes are still huge. So now, why do we care? I mean, we've heard this morning why we should care, but just to sort of expand on that, imagine you have 2 connectomes. One is a control and one is an experiment, and you want to know whether the difference in edge weight you see is actually true or just sort of chance. Same for example, if you were to compare female and a male brain, which is actually about to happen. The FAFB image volume is a female, and at Janelia they're currently proofreading a male brain. And then there's people that are interested in sexually dimorphic circuits, right? So how do you figure out whether the differences you're seeing aren't just bad luck? Okay, so what are the potential sources of variability? So one is neurons could be proofread to different degrees, right? So whatever, in dataset A, people just didn't put as much effort into proofreading neurons, so they're just overall less complete. The synapse detection introduces false positives and false negatives, right? Precision recall rates. You may have developmental abnormalities. And actually, even there, we kind of know that, what was it, like 0.5% of neurons have some weird thing going on. It's not a whole lot, but still. Or it's just biological variability, right? And again, to give you an intuition, so we've already looked at the synapse, right? But Vivek introduced that. But insect synapses are polyadic, so you've got one presynaptic site and then multiple postsynaptic sites at that synapse. What you see on the right are the precision and recall rates for the hemibrain for pre- and postsynapses, and each of these little bubbles is a brain region. And you can see presynapses are easier to detect. It's just the way it is. Postsynapses are harder to detect. The important thing is that precision and recall aren't 1-1, right? So, the purple points are the means. But there's also a huge spread across neurapils, right? So depending on where your connection is, you might end up having different synapse detection just based on the locality. Proofreading, so this is then the next step. What you're looking at here are the proofreading rates, so how many, right? So after the synapse detection, you've got free-floating synapses in this volume. And through segmenting the neurons, you sort of associate those synapses pre and post with neurons, which then ultimately make your graph. And you can measure this as fraction of synapses, pre and post synapses, that are associated with one of the 139,000 neurons at the end. And again, pre-synapses look pretty good. So this comparison is between hemibrain and FlyWire, actually. And each dot is still a neuropil. Pre-synapse pretty good. That's because the pre-synapses are typically on the big backbone. It's different from vertebrate neurons. So big backbone segment well, so easy to capture. Post synapses on the fine dendrites, not so much. So that you can see, like at worst, about 25% of the actual connectivity has been captured in this connectome. These are the give or take final rates. The needle is not going to move much more. Certainly not for the hemibrain. Right. So the connectomes that we're looking at are actually the sort of the sum of all these processes, right? So we start off with some probability that A and B are connected when you build a brain. So you use that to build a brain. That's where your biological variability comes from. And then we've got a sort of somewhat black box. Some of these things we know, but a black box where between the brain and the connectome, there's a whole bunch of processes that all introduce technical noise, or what we call technical noise. But again, we kind of know for some of these how much of an impact they could have. So what we did is we built a crappy little model. I'm saying crappy because that's a biologist trying to do math. It should be the other way around. But essentially what we did, we took the observed connectome, so our partially proofread connectome. We extrapolated a fictive ground truth, so the original brain from it. And then what we did is we essentially generated draws from that, where in each draw you remove false negative synapses, so synapses that the detection missed, added false positive synapses that and so on, and also randomly dropped synapses according to the completion rates of the neuropils. And you can do this twice independently, and then you end up having two connectomes that you know are from the same original connectome, and you can start comparing. And obviously, you can do this a couple hundred times to bootstrap the whole process. That's what you get. So this is essentially the same graph as before, but this is now if it was only technical noise. And you can see that the envelopes are smaller, they're still present, and we didn't factor all the possible sources of variation in. Right? For some, we just don't know what to do. How to assess this yet, or at least we didn't try in this model. And it looks like edge weight difference of about plus minus 30% could be purely based on the proofreading status and the synapse detection. And I think sort of moving forward, that's something that we as a field have to start thinking about a bit harder. And clearly, that model has room for improvement. So I invite you to start thinking about it.
So could you say again how you chose how much noise to inject into your
sorry. So basically, I used the proofreading rates. So that's not true. Sorry. For the synapse detection, I'm showing you hemibrain data. For the FlyWire connectome, which what I just showed you, the technical noise model is based on. So we're basically comparing left and right hemisphere there. I used the rates that were in the original paper for the synapse detection, which aren't per neuropil. So it's actually on that end, it's already slightly inaccurate. But it's the best data we have on that end. And for the proofreading status, it's basically these data points, along the x-axis. Right, okay. I think I've got 10 minutes left, right? Cool, perfectly on time. Okay, so I'm gonna circle back to cell types. It'll be really short and sweet, I think. Mostly because we've actually covered a lot of the bases already, because I think the consensus, I don't have to convince people that cell types are useful, at least I hope so, right? So they make it easy to think of, the graph by simply compressing the data, right? So at least in some instances. And in our case, they actually help us link, are the means to link across data sets and modalities. The issue is always that it's really hard to define cell types. We've probably spent the last year or so thinking about cell types, and I never wanted that to begin with, but it feels like you have to eventually. If you want to get to the end, it's a necessary evil. And there's many ways of defining cell types. This is just illustrating one approach where you have some sort of distance metric, you run hierarchical clustering, you get a dendrogram, and then you have to decide what you cut to make your cell types. I suppose most of you will have done this at some point, right? But I hope you can appreciate that that's sort of it's not a very robust process. Robust in the sense that if 5 people did that, we might end up with 6 sets of cell types. Let me show you a different example and ask how you would cut that. So let's say we've got a dendrogram, and I'm telling you that these are sort of co-clustered neurons from 3 different data sets, and this is how they distribute. How many cell types would you say are in there? Like, where would you cut?
Can you use the probabilistic model and not cut?
Just, intuitively.
Intuitively, I would use the probabilistic model and not cut.
Okay, fair enough. If you can't do that, if you have to cut.
4.
5, 3.
4, 5, 3. Okay, I'm going to throw in 2, right? Because if you look at it, there's only 2. Basically, if you want to find the smallest cluster that still contains representatives from all 3 data sets, and there's a couple catches in there, right? Like, what if one unit is just missing, blah, 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 right? But on sort of a conceptual level, only 2 clusters fulfill that criterion. So essentially, what this sort of translates into is the definition of a cell type, at least for us. A cell type is a group of neurons that is more similar, right? For example, these 3, or the red one specifically, more similar to a group of neurons in another brain than to any other neuron in the same brain. And I'll leave it by just showing you that that works. So what you see here is sort of a cutout of a big dendrogram where this is NBLAST and connectivity combined. But effectively, what we did here is co-clustered all these neurons from 3 different data sets, and then just used this algorithm to define cell types. And it turns out it works really well. And essentially what this does is taking biological variability into account, which is something that you don't if you just sort of cut at whatever level or whatever height of the tree suits you. Obviously, there's better ways of doing it. I'm just, thought that was the sort of more obvious example of why that's a bad idea. Okay, that's already it. I've got a couple of spare slides, but just briefly to wrap things up, there's a new and shiny FlyConnectome that I hope some of you will find useful. If you have questions, do get in touch either with myself or with people at Princeton. Sven or Ari would be good people to ask, although Sven isn't at Princeton anymore, for the record. But, yeah, I hope you find this useful. Then second, one of the second take home is that insect brains aren't snowflakes, but there is observed variability that is due to both biological and technical noise, and we have to start thinking about that. And then last but not least, I know this was sort of a very short detour, but if you want to define robust cell types, you have to take individual variability into account. And with that, there's a whole bunch of people to thank, both in Cambridge, but also a lot of people in Princeton. First and foremost, Sven, who again spearheaded the whole FlyWire project, but also Marla and Sebastian, obviously. And yeah, if you have any more questions, I'm happy to take them.